So welcome back from the break. Hopefully you had a chance to take a quick break. Let's take a look at these concept check questions and then move on and start talking about vertebrates. So identify the 4 derived traits that are found in all chordates at some point in their life. So those would be, of course, the notochord, the hollow dorsal nerve cord, the pharyngeal slits, and the postanal tail. Those are the common derived traits of all chordates that define the clade chordata. So those are the 4 traits that you should be familiar with, and hopefully you can talk about them in a little bit more detail as well. Let's look at the other question. You're a chordate, but you lack most of the derived traits of chordates. Explain why this is so. Remember, you're not required to have these traits throughout the whole of your lifetime in order to qualify as a chordate. You need to only be in possession of them at some point in your life. So we as Homo sapiens are chordates. We're also vertebrates. But that means that at some point in our life, we do have to show the presence of these 4 chordate features. So you do have a notochord very early on in your embryological development, but it gets displaced initially by cartilage and then ultimately by mineralization. You do have vestiges of your notochord, however, in the form of the cartilaginous discs that separate each pair of vertebrae. So the so-called vertebral discs, these cartilaginous discs that provide buffering between the bony vertebrae, are vestiges of your notochord. Of course, you do have your hollow dorsal nerve cord, that's your spinal cord. At the anterior end, it enlarges to give rise to the brain. And then there are branches that lead off into the appendicular or axial part of the nerve system to provide communication to and from the brain and all parts of your body, including the appendages. You do have not formal pharyngeal slits, but something that we refer to as pharyngeal clefts. They're not proper slits in this region of what would be your neck. They're clefts, little pockets. But because we are not aquatic, we do not need gills. So these are not going to develop down the pathway towards gills. Instead, these tissues are co-opted to form other structures. So some of the derivatives of those pharyngeal pockets or pharyngeal clefts become the lower jaw and the upper jaw. And we'll talk more about that when we talk about the origin of jaws. Some of the tissue becomes features within your neck, like the larynx. The tubes that connect your inner ear to your throat, the eustachian tubes, are derived from the pharyngeal clefts. And the little bones that help link your eardrum to your cochlea, the organ that converts the vibrations of the eardrum into nerve signals that carry the sound, the nerve signals of sounds to your brain, are also derived from tissue that originates in these pharyngeal clefts. And then we talked a little bit about the postanal tail, the fact that you do have a tiny little tail for a brief period of time, it's usually around 3 to 6 days before it gets reabsorbed again. And you have a vestige of that tail in the form of the coccyx, these fused bones that form the tailbone. They're actually fused vestigial vertebrae that would be part of the tail if we had a tail. So you do have the derived traits at certain time periods during your embryological development, but you lose most of them. The only one that you retain into adulthood is the hollow dorsal nerve cord. The others get redirected, or the tissues that would develop into those traits get redirected and reused and adapted to new uses, as evolution is wont to do. So that's the explanation for that question. So what is a vertebrate? Well, a vertebrate has a head, something that no chordate has. And so one of the things that you start to see with the head is the movement of sensory organs into a formal structure that initially is just an enlargement at the anterior end, but in vertebrates that have a more formalized vertebrate form, you actually see the presence of a cranium. So some of the very, very earliest vertebrates and one extant clade of vertebrates are more precisely referred to as craniates, although that is a term that appears to be falling by the wayside, but it is a term that has been used previously to describe the very, very basal extant vertebrates and also the very earliest vertebrates in the fossil record. They don't have a formal vertebral column in the strict sense of the term, but they do have a initially cartilaginous plate that gets displaced with bony material. That's the cranium that covers the brain and is the beginnings of the skull. So that's what we see as the very, very earliest evidence of vertebrates, the origin of the cranium and the craniates, a term that we don't use very much these days. So one of the things that separates vertebrates from invertebrates is the number of cassettes or clusters of Hox genes. So you may recall when we were talking about the invertebrates, both in chapter 32 and in chapter 33, that I emphasized the fact that all invertebrates have a single cluster or a single cassette of Hox genes. This includes the invertebrate members of the Chordata, the phylum Chordata. So the lancelets and the tunicates, like all other invertebrates, have a single cluster of Hox genes. All vertebrates, however, have two clusters of Hox genes. In parallel with that, we also see a duplication of another family of genes called the DLX genes, which are transcription factors. So Hox genes are transcription factors, but they are transcription factors that control hundreds of genes and lay down the body plan. The DLX family of genes are not quite as I need to backtrack on that. The DLX family of genes don't control as large a suite of genes as the Hox genes, but they are important in regulating genes in tissues. And so the duplication of these provides a increased flexibility in the utilization of the basic set of genes that are at the heart of all animals. We've talked about this before, the fact that there is a basic genetic recipe for animals, and all animals possess this basic set of genes that make up the recipe for animals. And it's not the number of genes that you have, it's what you do with them. So having more control genes in the form of the Hox genes and in the form of these DLX genes gives you the opportunity to create a more complicated and more diverse body plan, simply because you can utilize the basic recipe or the basic ingredients of the recipe in a greater range of applications with much more flexibility. And so you see the emergence of the vertebrate body plan as a consequence of being able to do more with the basic animal ingredients because of the increase in the number of control genes. So the defining features of vertebrates are the vertebral column, which encapsulates the spinal cord, a skull at the anterior end of the vertebral column, and in aquatic forms you have fin rays. In terrestrial forms you have limbs, and these are mineralized structures. So fin rays are actually bony material, but they are only distantly related to the bones that would become the basic form of the tetrapod limb structure. And you may recall that that is one big bone, two big bones, cluster of little bones, digits, the common form for terrestrial tetrapods. So one of the other features that we find unique to vertebrates is the fact that when the vertebral column starts to condense out, or more precisely, the neural tube starts to condense out. On the dorsal side of the neural tube, you have this cluster of cells that we refer to as neural crest cells. And these neural crest cells then migrate from their original location forwards, so anteriorly, so they move forwards to the anterior end, and they give rise to structures within the skull. And so you can see the derivation of the skull as part of the derivation of the vertebrate body plan, because the neural tube is something that is unique to vertebrates. It gives rise to the central nervous system. And the neural crest cells that condense out at the same time as part of the condensation of the neural tube from the epiderm, from the ectoderm, not epiderm, ectoderm, also contribute to the formation of the skull. So that's the reason why the central nervous system and parts of the skeleton are associated with the ectoderm when we're thinking in terms of the three germ layers and the systems that are derived from them. The neural crest cells are derived from ectoderm, and they give rise to parts of the skull. And so there is a contribution to the skeleton from the ectoderm under those circumstances. So here you can see the formation of the neural tube. Here are the neural crest cells that condense out at the same time from the ectoderm, and they then migrate forwards to give rise to structures within the skull. And you can see some of the structures here in a primate skull that are associated with neural crest cells. In the case of the vertebrates, we also know that the pharyngeal slits or pharyngeal clefts are no longer necessary for food filtering, because vertebrates eventually develop jaws and teeth to break up food. And so there's no requirement now to filter the food particles out of the water. So the pharyngeal clefts and slits and the supporting structures associated with them can be adapted to a new use. So their initial use is as gas exchange organs. We call those gills. This is important because we know that vertebrates are very mobile. They have a much higher metabolism as a consequence. So they need a better gas exchange system. So the gills provide that for the aquatic vertebrates. They need a much better circulatory system to carry oxygen and nutrients to all parts of the body. So they have a closed circulatory system. They have a heart to pump the blood because it's now legitimately blood in a closed circulatory system. So they have a heart that pumps the blood through the body. It only contains 2 chambers, and we know that the oxygenated and unoxygenated blood do mix within the heart as a consequence of this, so it's not as efficient in terms of maintaining separation between oxygenated and unoxygenated blood as we are familiar with in our own circulatory system, but it nonetheless serves the purpose of moving the blood efficiently around the body and allowing for oxygenation of tissues and also transport of nutrients. We see the rise of organs for filtering the blood. So we see the rise of kidneys for removing nitrogenous waste, and also the liver for filtering out other waste products and detoxifying the blood. And in the case of vertebrates, when you start looking at the liver, you find that one of the largest organs within the body cavity is the liver itself. It can be quite large compared to other organ systems, which is a reflection of the importance of the liver and what it does for vertebrates. So when we look at the most basal of vertebrates, we find 2 clades, Myxini and I've forgotten what let me take a look, hagfish, yeah, Petromyzontida, that are referred to as jawless vertebrates. So we typically think of vertebrates as having jaws, but the earliest vertebrates did not have jaws. And so we need to make sure that we understand the distinction. Jawless vertebrates have a cartilaginous skeleton as a consequence of lack of mineralization. And this is the early history of vertebrates. The initial vertebrate body plan does not include mineralization of a skeleton. In other words, a calcified skeleton. It's a cartilaginous skeleton. And so the early skull structure and the supporting structure derived from the notochord that supports the body is actually cartilaginous. And in the case of the hagfish, does not even provide any coverage to the nerve cord. So when you take a look at the hagfish, which are the most primitive, or most basal is probably a better term, of the true vertebrates, they look very, very primitive. They are sometimes referred to as slime eels, but they are not eels. Eels are fish. They are bony fish. These are not bony fish, but they're referred to as slime eels because they possess this row of protective slime glands. And they secrete this slimy material that expands rapidly when it comes in contact with seawater. And this will clog the jaws and the gills of any bony fish that is game enough to try and use them as a food resource. And so this is a protective device, because the hagfish do not have jaws. They do not have solid parts to protect themselves. So they have to protect themselves some other way. You can see the mouth structure here. It's unfortunate we can't see the pharyngeal slits, but the pharyngeal slits are laid out in the body down here. Let me put that in. So the pharyngeal slits, so this is where the pharyngeal slits are located, down in this region of the body. They either suck in water, or they suck in mud and sand from the ocean floor to process the organic matter that's in there. The food particles are filtered out and processed through the digestive tract. The water exits out through the pharyngeal slits, so the water comes out this way. So water in and water out to allow the food particles to be trapped. These are craniates, so they don't have a proper vertebral column, but they do have a plate of cartilage that sits in here over the very primitive brain that they have at the anterior end of their central nervous system. It's an enlargement of the anterior end of the central nervous system. It's a bit more complex than what we saw with the lancelets, but not by much. They do have a postanal tail. You can't see it because of the way the animal is curled up here. What is interesting is that people are now starting to eat hagfish in various parts of the world. So people are starting to see these as a potential food source. And when they are trapped and pulled out of the water in fish nets, they do secrete the slime, and you can see the action of the slime because it's still in contact with salt water, and it does expand and it creates quite a mess. It is a very, very good protective mechanism for these animals. The other group of jawless vertebrates is a little bit more evolved in terms of its interaction between the notochord structure and the central nervous system, because there are extensions from the notochord, cartilaginous extensions from the notochord, that partially wrap out and partially wrap around and enclose the nerve cord. It's not a full encapsulation as we recognize in vertebrates, where the vertebrae form a solid capsule around the nerve cord, but it provides partial protection to the nerve cord. Lampreys are born in freshwater streams and lakes, and as juveniles, they are suspension feeders. So they utilize their pharyngeal slits, again, for food filtering. Those that remain in freshwater remain as filter feeders and do not feed once they mature. They reproduce very quickly once they reach adulthood in freshwater, and then die off pretty quickly once they reach adulthood. Others migrate out to the ocean or migrate to lakes. We do have lampreys in the Great Lakes across the Midwest. So the Great Lakes have lampreys, and they are considered to be a nuisance for the fishing industry because they are parasites of bony fish. And the way they do it is to use this sucker disc device with these hardened teeth-like arrangements. They're not actually teeth, but they look a little bit like teeth. They use these to attach to the skin of their prey fish, and then they rotate the mouth structure backwards and forwards. So they rotate it like this, backwards and forwards, and rasp away at it, and also use the tongue, which you can see here, to rasp a hole through the flesh of the fish. And then they suck the blood and the interstitial fluid and any other loose material that they can out of the body of their prey fish and filter that out. And you can see here the pharyngeal slits forming a line down the body of the animal. So the food particles are filtered out. The other material is then released through the pharyngeal slits. And so you can understand why these animals would be considered to be at least a nuisance, at best a competitive parasite for the fishing industry. And so the animals that enter the ocean or enter larger bodies of water, like the Great Lakes, become these parasites. Eventually, they will return back to the streams for reproductive purposes, and then they die off in the stream. So these are the two clades of jawless vertebrates that are extant today. And we know these are the origins of vertebrates as jawless vertebrates, the craniates, something that we don't, as I said, not a term that we use very much these days, but is a term that historically has been used to describe these jawless vertebrates that don't have a formal mineralized skeleton, but do have the cartilaginous plate that covers the brain and represents the beginning of a skull-like feature, the cranium. So when we start thinking in terms of something that is more along the lines of what we think of as a vertebrate, the earliest that we can find in the fossil record, again, comes from the period of the Cambrian explosion. It's a little creature called Haikouella. It was represented in the painting that we saw back in chapter 32, I want to say, of the typical ocean scene or aquatic scene in a Cambrian ocean. Haikouella is the little bilateral creature swimming above Anomalocaris, the large arthropod predator. In fact, Haikouella is quite small. It's only about an inch long, but it does represent the beginnings of the vertebrate body plan. It is bilateral. It does have what appears to be a brain, eyes, muscle segments, but not a skull. So it's not a cranium, but it's showing the beginnings of the vertebrate body plan. Then we saw Mylocunmingia previously in the presentation. It does have a skull. It is considered to be a true vertebrate in every sense of the word. You can see in the picture, if you want to go back and take a look at it, evidence of vertebrae-like structures. So it looks like it has the beginnings of a spinal column, but again, it's still quite small, comparatively speaking. So this is Haikouella, and you can see that it has a bilateral form. It's got segmentation, it's got the pharyngeal slits, it's got an eye, it's got the sucking mouth for either sucking in mud or food particles out of the water, the pharyngeal slits for filtering purposes, but it doesn't have a skull. But it does have the appearance of the vertebral body plan. The first evidence of mineralization is through a group of organisms known as conodonts. And they are named after the mineralized structures that are found in the mouths and the pharynx of these organisms that are known as conodonts, or sometimes referred to as dental elements, but also referred to as conodonts. So this is an artist's representation of one of the earliest members of this group of organisms. And you can see these dental structures, these conodonts that are inside the throat and a little bit further down into the pharynx that allow them now to break up larger food particles into smaller particles. The appearance of these structures removes the requirement for filtering. So these animals are more aggressive in terms of their ability to access food. They are probably some of the earliest predatory animals because they now have the ability to ingest larger food particles or maybe even whole small organisms with the ability to break them up and grind them up using these calcified elements in their mouth and in their throat. These are not true teeth in the sense that we understand teeth, and these are most definitely not jaws, but they are the beginnings of calcification. They are beginnings of mineralization of the skeleton. These guys still have cartilaginous skeletons, however. In other organisms that are extant at the same time, you see mineralization taking place on the outside of the animal. So these are still jawless. They have mouths, but they don't have jaws in the sense that we think of them, but they have armor plating in the form of calcified plates on the outside of the bodies. And this also speaks to the fact that we have an increase in predation. And so it is likely that animals like these early conodonts with their dental elements are the sorts of predators that these armor-plated jawless vertebrates are protecting themselves against. And so the beginnings of calcification, the beginnings of mineralization does not lie with the internal skeleton. It lies with the feeding apparatus in the throats and the pharynx of the conodonts, and also in the presence of the armor plates on the outside of some of these jawless vertebrates that are cohabiting the same environment as the conodonts, and likely represents an attempt to protect themselves from the conodonts as more aggressive predators. So those are the very earliest of the vertebrates that we find in the fossil record, and also the most basal of the extant jawless vertebrates. Remember, all of these animals right now lack that feature that we associate with most vertebrates, the presence of jaws with teeth. These are all jawless vertebrates. We will address the origin of jaws in the next section. But first, let's take another break. And again, I'm going to make this a formal break. Here is our concept check question for this time period. How do the differences in the anatomy of lampreys and conodonts reflect the feeding methods of these animals? I'll see you on the other side of the break with the answer.